Okay, so evolution. Who's excited?
Woo!
All right, I am very excited too. You ready?
Go.
I believe in evolution.
I'm out.
Taking my
Craziness,
cake.
right?
Jacob,
Sit
crank the
down.
car. Crank the car, sir.
What's the question that should come after that? Somebody says they believe in evolution.
What type?
What's your definition?
Exactly. What's your definition of evolution, right? So the definition that I would apply to it is change over time, right? That's basically what the word means, is to evolve, to change over a period of time. And so I believe that to be true because it's observable. We can see change in people over time, right? We can see change in organisms over time. And so I believe that, and any God-fearing Christian that knows about this stuff and has read any of this kind of stuff believes in that, right? So, but there are those Christians that are staunch against anything that goes against the Bible, right? So they see things as black and white, but I'm here to tell you that change is happening. But the problem with evolution is that scientists, particularly secular scientists, have hijacked it, right? And so now it means change over not just a period of time, but over millions and billions of years, right? But we shouldn't be scared of change, and we shouldn't be scared of studies that look at change because it's part of God's design. And when God created all the different kinds, He put in these different variations and allows for us to have a bunch of different types of dogs, a bunch of different types of cats, right? And so it's good, and it's observable, so we can trust that it's true because it's observable. So I just want to start off by looking at Genesis 1:24 and 25, where God made all the animals. So it says, "Then God said, 'Let the earth bring forth living creatures after their kind, cattle and creeping things and beasts of the earth after their kind,' and it was so. God made the beasts of the earth after their kind, and the cattle after their kind, and everything that creeps on the ground after its kind, and God saw that it was good." So we see in Genesis that God creates kinds. Does anyone have any idea exactly what that means? So we have these classifications, right? Kingdom, phylum, class, order,
Family.
genus, family, genus, species, right?
Yeah.
So where do you think kinds falls in that? Any guesses?
That species.
Species is the lowest.
It's the lowest.
So that you have the greatest amount of variety there. So it's higher than that.
Family.
Class.
It probably happens at a familial level, right? So a family. But the thing about that you have to remember is that it's not hard and fast, right? Because man came up with this classification system. So just because we say this is how it's classified doesn't mean that that's the way it is. And there's some variety. I mean, we don't know everything there is to know about how God set up kinds. We just have this limited information in Genesis. But at the same time, we can see that there is variation. So we don't have to have, God didn't have to create every species the way it is. They can evolve from their specific kind, okay? So one thing though that I thought about as I was thinking about this is, what if the fall didn't happen? What kind of variation would we have, and what implications would that have for species and development? It's kind of weird to think about, right? Because we think of change over time, and we'll get into that. A lot of that is associated with mutation and bad things and loss of genetic information. But God designed it to be there, for there to be variety. And so some of that variety isn't a result of the fall. A lot of it is, right? So a lot of variation, a lot of changes have to do with the fall and the fact that we have mutations and diseases and stuff like that. Don't get in the video when you pick that up.
I'm staying over.
Okay. So I don't know the answer to that, like what would variation look like if there wasn't a fall, but I just thought that was a weird thing to think about. So the first thing I want to introduce tonight is confirmation bias. Who's ever heard of confirmation bias? Yeah. So this is really the crux of this whole issue of evolution. And confirmation bias, the definition is, it refers to the tendency to selectively search for and consider information that confirms one's beliefs. Okay? And so what that means is that if someone wants to discern whether or not something is true, they already have this idea of what's true before they start, right? And so this preconception is going to point them in a direction of finding things that support their idea. And then if you find something that supports your idea, that gives you more reason to believe your side of whatever issue you're on, right? And so, but this doesn't really leave room for alternatives. And if you really don't have any basis for what you believe, even if you find one piece of evidence for what you believe, then your wishful thinking that that's true is going to grow, right? And the whole point of that is that the person that's practicing confirmation bias, and we'll see that pretty much everybody practices it, right? Everybody has these preconceptions about whatever they believe, and if you find something that confirms that, you're going to believe it even more, right? But the issue with confirmation bias is that people are scared of being wrong more than they want to know what the truth actually is, right? So you have to, somebody who practices this, they have to want to know the truth. They have to want to believe that there could be an alternative to what they believe before you can even work on trying to convince them of a different truth, right? Something that isn't what they're believing. So just a couple of examples of this. We talked about, when we talked about carbon-14 dating, Dr. Jack Horner, which is one of the scientists who worked with Mary Schweitzer on doing different dinosaur bone and tissue experiments and stuff. So he had a phone interview, an impromptu phone interview. This radio station called him and said, hey, it was like a Christian radio station, if we give you $20,000, $30,000 to conduct carbon-14 dating on these dinosaur bones, would you do it? And he said no, he wouldn't do it. If they came up with the money to pay for it, he wouldn't have to pay for anything, then he wouldn't do it. And the point of that is, if he truly believed that there shouldn't be any carbon-14 dating, why not do it, right? If there shouldn't be any carbon-14 left in those bones, just do it, prove them wrong, and then move on, right? But he was so afraid that something could come up and that the Christians could twist it in a way that would support creationism, that he wasn't willing to be a scientist, right? Because that's what science is all about, is trying stuff out and trying to disprove something. But the interesting thing about confirmation bias is if you type in confirmation bias in Google and you go to Rational Wiki, I don't know if you guys have ever visited Rational Wiki. It's kind of like Wikipedia, only for like logic stuff, and it's very heavily atheistic. But one of their examples of confirmation bias is Christians and creationists who believe in the flood. And so Christians and creationists who see fossils buried in rock layers laid down by water all over the earth, that's a quote from Ancient Genesis, but that indicates to them that there was a global flood, right? Because we would expect to see all these dead things buried in rock layers all over the earth, and that's what we see. So atheists point to that as confirmation bias of Christians because, oh, you see that, so you think it's this way because that's what supports what you believe. So, but all that to say is that, I've said this before, we all have the same evidence, right? So we all look at the same earth, we look at the same fossils, we look at the same samples, and it's up to the individual to interpret what that really means, right? So it either supports you or it doesn't, but you can twist it to support whatever you really want to do. But this leads us to an interesting thing that happened in the Ken Ham Bill Nye debate. One question that was posed to Ken Ham was, what, if anything, would lead you to believe something else? Well, what, if anything, would change your belief in God? To which he replied, I can't, right? Because his belief starts with the Bible first, and it literally doesn't matter what anyone else says, what science says, what Bill Nye says. You know, he starts from the Bible, and then anything that contradicts the Bible, in his mind, and in my mind, it's not completely fully understood by us. And so it's up to us to wait to see, you know, if there's different interpretation that aligns with the Bible, because this is what we start with, right? And so he got slammed for that because he's not open-minded, right? But at the same time, we really can't be, because we learned in the first lesson that, you know, what is your ultimate source of truth? If it's people, people are fallible, people lie, people steal, people cheat, right? And so I don't really want to put my ultimate source of truth in a person who does bad things, right? I want to put it in something that is true, completely true, and that we can know is true. So everyone has confirmation bias, but it's good to be aware that it happens, so that possibly, you know, we can be more aware if we're practicing it too much, right? But at the same time, you can't convince anything of something unless they truly want to believe that there could be something else that's true. Okay, so that's kind of the backdrop of what we're going to get into tonight, is looking at different evidences of evolution and looking at how it can either support people who believe in macroevolution, molecules to man, or it can actually support a creator God that made everything and set this all up in his design. Okay, so the first thing is we're going to go through a quick history of DNA, just to give you an idea of really how little we know about DNA. You know, we think that we know a lot of stuff about cells and how they function and all that, but we're still learning a lot, and there's still a lot of stuff that has yet to be discovered that's probably going to change the course of what we know about how a cell functions. So in 1830 is really where it all got started, when Charles Darwin went to the Galapagos Islands, and he went there on this voyage. He didn't go there to study animals, but it was kind of like a side thing that happened while he was there. And so he saw these finches, right? And so we've all heard Darwin's finches and how they have different beaks, and that means that they evolved from a single bird, which makes sense, right? We can agree with that, but that he actually took this information and then extrapolated it back and said, you know, if we can follow that these birds probably descended from one bird, then that bird probably descended from something else, and then he tried to postulate that it goes all the way back to a single cell, and then from that everything came to be, right? So he did this research in the 30s, and then in 1859 he published his book, which had all this in it, On the Origin of Species. In 1866, a monk by the name of Gregor Mendel. Does anybody know who Gregor Mendel is?
The geneticist.
He is the father of modern genetics. So Gregor Mendel worked on pea plants, and so he bred these pea plants, and he was able to show that different traits of these pea plants were inherited together. And he was also able to show that there was different ratios of how these traits were inherited. And so he really laid the groundwork for understanding how traits are inherited and how these different ratios help us to be able to predict how these traits are going to be inherited. But at this point, it's important to note that nobody had any clue what DNA was. Nobody had ever discovered it before. Nobody knew what genes were. So they were looking at simply traits that you can see, which were related to the genes that were in the DNA. So it's incredible, actually. Like, anybody who ever talks about him just says that it's just incredible that he was able to do this research, because there's no basis for it, right? It was just observation and pea plant after pea plant crossing and growing, and I mean, it's long, laborious work, but he laid the foundation for everything we know about DNA now.
When did he do that?
1866, around there. In 1869, DNA was finally discovered, so a little bit after Gregor Mendel did all of his stuff. But just because it was discovered doesn't mean they knew anything about it, right? So they discovered this mess of stuff in the cell, and it wasn't until 1952, so almost like 90 years later, 80, 90 years later, that we figured out that DNA is what housed genes and what gave rise to different traits and carried the information for the cells in the body. And then in a year later, in 1953, the double helix was found. And so it's pretty cool. The two men that have credit for the experiments are James Watson and Francis Crick. There's another lady, Rosalind Franklin, who did a lot of the actual experiments. She got, because they did it with X-rays to figure out that there's double helix, so she got a lot of really bad cancer and stuff from that. So then she doesn't get, now she gets a lot of the credit, but originally she didn't get a lot of the credit. But James Watson was actually, he started the place that I went to in New York. So he started that lab, and then he actually still lives there, and he like walks around campus and he drives a Jag, and so our class, yeah, well, he's kind of a grumpy old man, but he drives a Jag around, he eats food wherever he wants, but our class got a picture with him at the end of our time there. So, but it's just kind of cool to see like the guy who did all the work and stuff. So it's pretty cool. And then there's a lot of different stuff that happened between 1953 and present day. They discovered genes, obviously, and they discovered some types of regulation of these genes. In 2007, I believe, or in 1990, they discovered what are called microRNAs, which I kind of work with a little bit, but they're just another level of regulation of DNA that they discovered after they thought they knew everything about DNA, right? So there's stuff that we probably don't know about DNA still that we'll find out eventually. 2003, the human genome was sequenced, right? And that was a big deal because that opens up the door to personalized medicine, and that's eventually where you're going. What I think is going to happen is you're going to go to the doctor, and you're going to have your genome on file, and then the doctor can look at your genome, look up whatever he needs to look up, to how you're going to react to drugs and how you're going to react to different treatments and stuff. So, I mean, it's going to be amazing once we figure out exactly what to do with all this information. Because if you sequence the whole genome, that is a ton of information. And the problem right now is that, honestly, we don't know what to do with it. So people are working on trying to figure out how we take all this information and condense it into a form that physicians can use and doctors can use and stuff like that. So that's where we are right now with DNA, is we're kind of in this limbo of trying to figure out how to use this abundance of information that we have. So now we're going to talk about DNA. Get down to some specifics. How many base pairs are in DNA? Does anybody know? There's going to be a lot of quizzes here.
23.
So there's 4 different bases. And so our genome is comprised of 4 letters, which is incredible. And so the combination of these letters are what give us the DNA code. So there's A, T, G, and C. So, and there are 3 billion of these in everyone's genome. So there's 3 billion of these letters lined up, and they're in different groups, right? They're in different chromosomes, but there are 23 chromosomes in a person, and those 23 chromosomes end up housing these 3 billion base pairs that have all the information necessary to make every single cell in your body. Now, the interesting thing is that we have different kinds of cells, right? So we have eyeball cells, and we have foot cells, and we have all these bone cells, right? So if each cell has the same amount of DNA, the same exact DNA, then there must be something that regulates expression of genes that lead to a certain kind of cell type, right? So it's just, that's kind of what we're working on still too, is how to understand that regulation. And it's crazy. That's the kind of stuff that we're doing right now. Okay? So out of 3 billion base pairs, only 1% of that 3 billion base pairs, which I should have wrote down how much that is. So that's like 30 million. 30 million of those base pairs are what code for genes, what we know as functional proteins within the cell. So that means that 99% of your genome doesn't do anything, quote unquote. So that's what was thought once we figured out how many base pairs there were, is scientists said, okay, so now we've sequenced the genome, we've found all these genes, and now there's all this DNA that doesn't get used, so it must be junk DNA, right? And so we'll get more into what this means by junk DNA and why it's actually not junk DNA here in a little bit. Does anybody know what happens to the DNA? How do we read DNA? This is the molecular biology, the central axis of molecular biology. So we start with DNA. And then this gets read by proteins, and it gets transcribed into RNA. Okay? RNA. So this is in the nucleus. Okay? So if the DNA is in the nucleus, but it gets made into functional stuff outside the nucleus, this is the message that takes it from the nucleus out into the rest of the cell, where it can become protein. Okay? And then proteins are what go all around your cell and do all the things that make your cells work. So there's regulation throughout this whole thing, but a lot of the regulation that gives rise to differential cell types happens here. So you're only going to make RNA for proteins that you're going to use in your cell, right? So before we even make RNA, we're going to regulate which genes are expressed in that cell. Any questions? I'm just trying to give a basic background for DNA stuff, because this is what I do, so I'm trying to let you in on my life a little bit. Yeah, I'm going to need to talk about that. Okay, so yeah. So if we're reading DNA, what causes DNA to be read is,
Now you know what my life is like every day. What did you do at work today? Oh, you know.
science! Yeah! Lots of science. Okay, so if you look at different pictures of genes and stuff, this is a pretty basic picture of a gene. So if this is DNA, okay? So this is a DNA sequence, but it's pictured like this to denote that this portion of the DNA encodes for the protein. This has the information that matters and ends up being the protein, okay? So, but in order to express this protein, you have to actually have other proteins that come up here and bind to what's called the promoter, okay? So the promoter promotes the transcription or the reading of the DNA for the gene, okay? So this is how you get the regulation of which genes are expressed, because certain transcription factors promote certain genes, okay? So this is how we get the different expression of different proteins and stuff, okay? So this will be really important later when we talk about different examples, is that certain proteins regulate the expression of other proteins, okay? So we already talked about how we can observe that there are differences in people. You like that? Super marker. Pretty awesome. So we can observe that people are different, right? And the reason for that is because there are variations in genes, okay? So if we look at gene X in me and in Blake, it's probably going to be different. But it encodes for the same protein that does the same thing, right? So the reason it's different is because there's one base change. So if this top sequence is me, C, T, A, G, Blake's sequence might be C, C, A, G. Okay? And so that will change a little bit of how the protein works, which gives rise to Blake. And we all love Blake just the way he is, in spite of the fact that he's mutated from me.
That's true.
In spite of the fact that he's so different from me, we all still love it.
Thanks, man.
I love you, man.
Why gotta be mutated, man?
It's not a mutation, right? It's just a variation.
Which is
So each of these genes that has a difference is called an allele. So we have 2 copies of each gene, right? So if I have a second copy of this gene, one copy might be the way I had it on the top, right? But the other copy could be the different copy. So the alleles don't necessarily have to be the same within your genome. You can have 2 different alleles of the same gene, which gives rise to differential function of each protein. How confusing is that?
Can you spell that?
Allele?
Yeah. I don't even know where to start.
L-I-L.
Oh, thank you.
Is that right?
I think so.
Yeah, that's it.
owl.
Yeah, that's how you spell
Okay.
it. Didn't look right when I wrote it.
There's no Q in it.
So, one way that people like to claim evolution is true is to actually look at these different alleles. And so you can sequence people's specific locations in their genome, and you can come up with differential alleles. And so in populations of people, you can track how alleles are propagated, how they're spread through different subpopulations of people. And so, you know, you can see kind of how people spread out, which is true, right? We all started from 2 people. And over that time, we changed and we became, you know, different ethnicities, right? And that's because of genetic variation. Now, if you have interbreeding of the same ethnicity or the same group of people that departed from the Tower of Babel, right? Then those people are going to have the same genes that get passed around. And they're going to have new genes, new alleles, sorry, I should say, new alleles, not new genes, that get passed around their group of people. But a different group of people isn't going to have those alleles. And so that's kind of one way that people say that evolution is true, is because they can sequence people's genomes and track these alleles when they pop up, how prevalent they are, and they can see if they're in a different ethnicity. So if 2 ethnicities share the same allele, then that means that they probably derive from the same group of people, okay? But the thing about that is, that aligns with what we believe. We believe that God made Adam and Eve, and from that, everyone else came, right? So everyone we should be able to track how people developed, how people changed, how people moved around the planet from the Tower of Babel. So tracking these things and doing population genetics, which is what it's called, is okay with us, right? Because we believe all the same stuff. So the problem is, what came before Adam and Eve, right? Was that chimpanzee or was that God? So that's where we get into this issue of confirmation bias. Is we both have the same information. We can all track alleles, and this is true. But the problem is how you extrapolate that. Do you go back before Adam and Eve and say, okay, we can track it in humans, we can track alleles in humans, we can track alleles in monkeys, so there must be a common ancestor for those, right? That's not true. It's not observable. We can't confirm or deny that because we can't go that far back. So we have to have some other source of truth that tells us whether or not we descended from monkeys, and that's what this is here for, right? It's God being very nice to us and explaining to us exactly how it happened, so we don't have to run into this question of why do we observe this? We observe it because it's from Adam and Eve. Okay? That being said, so does anyone know who our closest relative is from the monkeys?
The Nobo.
I think that's higher up.
Oh.
What? Come on, different types of monkeys. What we got?
Oh.
Baboon.
Baboon.
I look like a lemur.
I look like a lemur.
But kind of wiry. I hear you like to move it.
I do like to move. So, people say, secular scientists, evolutionists say that we are, have you ever heard 98 to 99% the same as chimpanzees? You ever heard that? What that actually means is that when you look at just the sequences of genes, we are 99 to 98% the same as chimpanzees. Which kind of makes sense because we have very similar structure, right? We have 4 arms and, well, 2 arms and 2 legs, 4 appendages, right? We have similar head structure, all that is very similar, so it makes sense that our genes look very similar. So there is this 98, 99% similarity, but that's only in the coding regions of genes, which is only what gets expressed. So the issue comes with if you sequence the whole genome, right? Because this is only 1% of the whole genome. So if you sequence the whole genome of humans and chimpanzees, there's actually only 70% similarity overall, which people don't like to say. Why?
Confirmation bias.
Confirmation bias, right? Because that doesn't line up with what they want to say about evolution. It doesn't line up with the fact that we are directly descended from chimpanzees and therefore we are very similar to chimpanzees. So it doesn't get publicized as much. But, so next time somebody says that, you're armed with the ammo to...
Full genome.
Yeah, exactly.
I've never heard that before.
Yeah. The other thing is that we are actually 90% similar in the coding regions to mice,
Hey.
which I bet you would have never guessed that. But so that's actually why we use mice a lot in research, is because it's very similar genetically to humans. So things that we find in mice, if we discover a function of a protein in a mouse, it's probably going to be very similar in humans. So that's why we use mice as our and they have very short lives, and so they're easy to grow and they're easy to utilize. And I know it makes Helen sad a lot. But next lesson I'll get into research. So, and I have something that I haven't even read anywhere else. It's just a thought that I had about research and animals and stuff. So stay tuned for the next lesson.
God, what, a month from now?
Which
Come
actually
on.
might actually just be evolution again, because I have like 2 more pages of stuff to get through. Confirmation bias. Okay. So another thing about how closely we are related to chimpanzees is, so they actually calculate how far back we diverge from chimpanzees. So the thought right now is that between 6 and 8 million years, which if you think about it, is a long error bar, right? So like 25% error involved in this, right?
Hey, that's
6 to
science.
8 million years. So they believe that we diverge from chimpanzees, and the reason they believe this is because of the mutation rate that is inherent in people. So each lifespan, a certain person is going to have, on average, about 60 new mutations in their DNA, in their coding regions, that affect their gene expression. Okay, and that gives rise to all this variety that we see. So if you have 60 mutations per, what did I write there? Per generation. And then you can multiply this by the lifespan of generation, which is years per generation. And you can look back and see, this is what they do to show that we're evolved from chimpanzees. So they look back and they see at what point, if we get rid of all these mutations that are different from chimpanzees, how long would that take to develop from chimpanzee DNA to human DNA? And so that's how we get this 6 to 8 million year backup. So there's no basis for this, right? Because we can't see it. We can't see that chimpanzees were chimpanzees at one point, and then a Neanderthal popped out, and then now we have people, right? We can't observe that. So this is all conjecture based on what we can observe. So we can interpret it in whatever way we want to, but this is how they calculate that. And I just thought it was interesting to kind of understand, because I think one thing that if you read an article about a science thing is articles simplify things a lot. And they take things that scientists say as absolute truth. So I think it's interesting to actually sometimes think how they come up with these different numbers and kind of be able to question that kind of stuff, rather than just glossing over it and saying, oh, I don't believe that, but really figure out why people think the way that they do. Okay? Yeah?
How do these mutations come about? Is it just like because maybe you got sick, or is it because that's just like how you're made?
So it could be sickness, but the real problem is that when you replicate your DNA, so every time a cell divides, it has to make an entire new copy of 3 billion base pairs, right? And we live in a fallen world, and so everything is not perfect. And so the proteins that are responsible for replicating your DNA make mistakes. So over the course of 100,000 base pairs that this protein is adding, it could make one mistake. Mm-hmm. And the thing about making one mistake is that there are really bad diseases that just depend on one letter being changed, which is unbelievable. And so we know now how important each letter in the DNA really is to the what's the word I'm looking for?
Integrity.
Integrity. Yeah. Integrity of the DNA. So, but the interesting thing is that you may have a mutation in your arm, right? You may grow a cancer somewhere, but at the same time, the only thing that really matters is your gametes that give rise to babies, right? So you may have a mutation somewhere else in your body, but that mutation won't show up in your child. So the thing about, like if you get sequenced to look at, what my child might have, like disease my child might have, so they're going to sequence cells that are going to give rise to children. They're not going to pull cells out of your mouth or something to go sequence, right? Because those cells aren't going to give rise to people. Your cells in your body are going to be different depending on where you sequence them. And so that's the important thing, is that the cells that matter are the gametes, so the sperm and the egg that gives rise to a kid. Does that answer your question?
Yeah, that's good.
Okay. What time is it?
22.
Yeah.
Oh boy.
Better listen fast.
Okay, we're gonna, can I have like 10 more minutes? Is that right?
Well, we didn't start till like quarter.
Yeah, somebody brought cake.
I think that
Thank
box is
you.
15 minutes.
Okay. So, I'm talking about natural selection first, for a minute here. Natural selection, has anybody got a definition for me? What do you guys think natural selection means?
The weak get thinned out.
Okay,
Survival of the fittest.
is it true?
To a degree.
Yes.
Yeah.
It's observable. We can look at this in a lab and we can see that it actually happens. So, and like what I've talked about before, it's what you do with that information afterwards. Do you extrapolate it back into molecules to main evolution, or do you extrapolate it back to God's design to give us, or the fall really, right? That's why we have natural selection in the first place, is because death was introduced at the fall. Without death, we wouldn't have natural selection, right? We would just have variation. So here's the definition that I got from Wikipedia,
Hey.
a great source of information on the internet. The thing about actually Wikipedia is that science stuff on Wikipedia is actually pretty good, because there are people that actually monitor those kind of pages and make sure that all that information is correct, and they cite papers and stuff like that. So just FYI. So natural selection is the gradual process by which heritable biological traits become either more or less common in a population as a function of the effect of inherited traits on the differential reproductive success of organisms interacting with their environment. Right, so that gets back to the whole idea that mutations and stuff don't matter unless they get passed on to the offspring. So, but there it says that biological traits can become either more or less common. So it's both. If you get a mutation in an allele that results in a betterment for the next offspring, then that trait is going to get passed on more. But if the environment changes and it selects for not needing a certain allele, then that trait is going to get lost over time. And that's something that we can observe. Here, there's a paper, I can't remember the year, but one of the big papers that came out had to do with bacteria. So bacteria grow very quickly, right? They have lifespans of as short as 20 minutes, or doubling times as short as 20 minutes. So that means every 20 minutes it gives rise to a new bacteria. But I don't know how large bacterial genomes are. That means that every 20 minutes, a bacteria has to replicate all of its DNA and make a new bacteria. So if you think about how many bacteria you can make over the span of about 20 years or something, that's a long time. So this experiment that I'm going to talk about has been going on for 20 years, where they take bacteria, they passage it, which means they just grow it, and then they freeze down some so they can analyze it, but then they just keep growing these bacteria and looking at different mutations that happen to try to track evolution, to try to see it happening as it's happening in the lab. So the experiment basically is we have E. coli, and these bacteria are marked by not being able to utilize citrate, which is a molecule that we use for metabolism, to make energy and to make fats and stuff like that, okay? So this is a molecule that's normally used in metabolism, in cells, in bacteria, in people, everywhere. So it's really common. But E. coli are characterized by not being able to use citrate in the presence of oxygen. So if there's oxygen present, it doesn't use citrate, okay? So, but over time, if you culture these E. coli on high citrate, in high citrate media, right, and you try to deprive these cells of glucose. So essentially, glucose is what they would normally use, right? Your cells are used to glucose, so that's what they prefer to use. Bacteria are the same way. But if you deprive these cells of glucose, this is your selective pressure, right? So this is how we get natural selection, is that we're changing the environment in a way that we're trying to make these cells evolve in a way that will adapt to the new environment. So if we decrease glucose, we have an abundance of citrate, then those cells that mutate to be able to utilize citrate in the presence of oxygen, those cells are going to do better, okay? So we have, they tracked it out, and over the course of 31,000 generations, which is passages of cells. So each generation has millions of different, not really millions, but X number of divisions of the cell. And then 31,000 of those division segments end up giving these E. coli that are categorized as citrate positive. That means they're able to utilize citrate. And so the way that this happens is that genes sometimes can move around the genome, and I don't know a lot about exactly how that happens, but if you have a gene, so let's say you have gene X here, and then you have gene Y. So in one cell, out of all these millions of different generations of these E. coli, it just takes one cell, and then that cell can give rise to new cells, right? So if you have one cell that moves the gene X under the promoter of gene Y, so that means that gene X is no longer controlled how it would normally be controlled, but it's now controlled the way gene Y would be controlled. And this means that gene X is going to be expressed differently than it would have been expressed normally, right? So that's how this ends up happening, is that these cells that have a citrate transporter that allows the citrate into the cell, that citrate transporter is now under the expression of the gene Y, okay? And so these cells now are able to import citrate into the cell and utilize it to give them a selective advantage, because that is the most abundant resource available in the environment. And now that we're able to import it into the cell and utilize it, then those cells are able to grow better. They outcompete those cells that do not have this mutation, okay? So this is natural selection. This is observable, right? We see this happening in the lab. This took 20 years. But this is the kind of experiment that shows that evolution, things changing, forms, things going from not being able to utilize citrate to being able to utilize citrate happens. But the key thing in all of this, and we'll get into this more as we get into different examples, is that what did this E. coli give rise to? What did it produce?
E. coli.
E. coli, right? We didn't see E. coli produce a bird, which that's a crazy thing, right? You can't be like, I want to see E. coli evolve into a bird before I believe evolution. But we see E. coli giving rise to E. coli that are a little bit different, right? So there's no new information happening here. It's just a reshuffling of information, but we still have the same organism that we're working with. But if you read this paper, then it talks about, hey, look at this cool thing that we did, and if you think about it, then this could probably mean that evolution's true. Or this shows that evolution is true because things change over time. Awesome. And but now people accept this. And so if you read a paper that talks about differences in genes changing, this is the conclusion they arrive at. And I really wish we could do more tonight. We'll have to save it for next time. Because the next thing is Darwin's finches. And we'll look at how finches make finches, right? Finches make finches, and E. coli make E. coli. They don't make different things, okay? And so, next time, we'll get into Darwin's finches. So, thanks for coming.